My name's Mark Gold. I'm the acting director of UCLA's Institute of the Environment and Sustainability. Welcome to the Oppenheim Lecture, the third one of our academic year with Paul Barber. Thank you for coming out tonight. I greatly appreciate it. We couldn't do this series without Tina Quinn and the Oppenheim family. They've been supporting this now for quite a long time. And also want to thank Kay Thomas for putting together the program for the year. Hopefully you guys have enjoyed it, and I'm sure next year will be also an extraordinary series. Also, Lee Cooper is here from our board, the Institute of the Environment and Sustainability, with Anne. Thanks for coming tonight. Also, this talk is co-sponsored by Ecology and Evolutionary Biology, as well as Atmospheric and Oceanic Sciences. So thank you guys for your support here. The talk tonight, as you know, is changing the fate of the Amazon of the oceans. Paul will talk extensively about that. Obviously, you see some pretty extraordinary visuals as I'm speaking, and a lot of concerns there with overfishing, pollution impacts, climate change impacts, destructive fishing gear, as well as using of dynamite, among other things, which I'm sure Paul will get into in depth. So we try to finish our series usually every year to have at least one local faculty member at UCLA, since we do such extraordinary work in the environment here at UCLA. And so we're glad to close the year with Paul. And really? Okay. I thought I was being really loud. I will eat the mic even more than I thought I was doing. All right. Paul has been a professor in ecology and evolutionary biology since 2008. He's a marine biologist that's focused on the evolution of marine biodiversity in the Coral Triangle, particularly in Indonesia. In addition, Paul founded the Indonesian Biodiversity Research Center that has helped build scientific capacity in Indonesia. Paul's also had extensive work in coral reef conservation within the region, particularly working closely with Conservation International. His academic background, he received his PhD from UC Berkeley. Strangely enough, we both started doing herpetology and ended up working in the oceans, which I find pretty interesting. He postdoced at Harvard and was a professor at Boston University before he came here to UCLA. Recently, he was a recipient of UCLA's Diversity, Equity, and Inclusion Award for his work on diversity project and peer program, among other things. He's also received UCLA's Distinguished Teaching Award. Perhaps most noteworthy for me personally, he made one of my dreams come true recently. My oldest son, Zach, will soon be a PhD student of Paul's in the fall. More importantly, my bills will go down substantially since he's coming from Stanford and going to UCLA. So that's just as important. So without further ado, here's Paul Barber.
Thank you, Mark, for that introduction. And thank you for the Oppenheim family for supporting this. And thank you all for being here. I always begin my talks with a bunch of thank yous, because without the work of a lot of really extraordinary people, none of what I will talk about today would ever happen. I've been privileged to work with really talented groups of students, both from the US and Indonesia at the graduate and undergraduate level. I have tremendous faculty collaborators, and there's a lot of students in my lab who are here tonight, and without their efforts over the years, I really would not have been able to accomplish even a fraction of what we have. I'd also like to thank UCLA, Conservation International, the U.S. Agency for International Development, as well as the National Science Foundation for funding. And last but not least is my family, who have been coming with me to the field since we met. since they've been born. They've always been there. They've sacrificed every one of their summers pretty much for their entire lives to be with us and to allow me to do the kinds of things that I do in Indonesia. So without their love and support, none of this would be possible. So we're going to talk tonight about an integrative approach to conservation in the Amazon of the ocean. And what does it mean, the Amazon of the ocean? Well, for some of you, you may be conjuring up something like this. I'd like to think that this is far-fetched, but you never know. Really, what I'm doing is trying to draw a parallel to the Amazon basin. And the Amazon basin is the largest, most biologically diverse terrestrial ecosystem in the world. And similarly, the Coral Triangle is the largest, most biologically diverse marine ecosystem in the world. It's defined by the presence of more than 500 species of hard coral. There are hard corals and soft corals. If you step on it and you say, ouch, it's probably a hard coral, although there's some soft corals that you don't want to step on either. It encompasses 6 million square kilometers, so it's about 2/3 the size of the entire continental United States. And it encompasses 6 countries, including the Philippines, Indonesia, Malaysia, Papua New Guinea, East Timor, and the Solomon Islands. Now, this is the most biologically diverse marine ecosystem on the planet, but what do we mean when we talk about biodiversity? Well, biodiversity is quite simply variations in life forms in an ecosystem, in a biome, the planet. We could look at biodiversity within this room. We could look at biodiversity on the scale of UCLA or Los Angeles, California, the United States. You just define an area and you enumerate all of the diversity that's there. Now, when we look at patterns of marine diversity, what we see is a striking pattern where going from high diversity, where it's red, to low diversity, where it's blue, this region of the Coral Triangle has the highest diversity in the world, and as soon as you move any direction away from the Coral Triangle, biodiversity decreases very, very rapidly. Now, we're talking about high diversity, low diversity. Well, what does this mean? So I'm going to give you some perspective. So let's just look at the Caribbean. So in the Caribbean, people have heard of the Caribbean? Everyone? Right? Okay, so there are 2 species of Acropora coral that occur in the Caribbean. These are the elkhorn and staghorn corals. And in comparison, if you look at the Coral Triangle, there are more than 100 species of Acropora coral. You can go to a single dive site in eastern Indonesia, and there will be more than 80 species of Acropora coral there. So we're talking about an ecosystem in the Coral Triangle that is 50 times more diverse for this genus than the Caribbean. So how many of you guys had heard of the Coral Triangle before tonight, before hearing my lecture? No, in the front, you're not allowed to raise your hands. No, that's cheating. Okay, so we have a handful of people that have heard about the Coral Triangle. This is the world's most biologically diverse coral reef ecosystem, most biologically diverse marine ecosystem, and most of the people in this room, in fact, most of the people everywhere have never heard of it. So we can talk about numbers, but better than talking about numbers is let's just bring you there. So we're looking at some footage from eastern Indonesia, a region called Raja Ampat, where there are tremendous coral reefs that support a great diversity of life of fishes and corals. There's actually over 1,750 species of reef fish, including really big things like those manta rays or these whale sharks. You get really interesting things like hairy frogfishes. So this is a type of anglerfish. And you've got this big individual here, and the little guy that's following it, that'll show up here in a second, that's not the baby, that's the male. So this is a mimic octopus. It's going to turn itself into a flounder and swim away. This is an octopus mimicking that same hairy frogfish. So just this tremendous diversity of life. These ecosystems are absolutely spectacular with vibrant colors and life everywhere. And the natural beauty of the place is astounding. And at one level, protecting that natural beauty should be enough. But we can think about this diversity in different ways. So the Convention on Biological Diversity states that 40% of the world's economy and 80% of the needs of the poor are derived from biological resources. Our societies are totally dependent upon biodiversity. And if we look at the Coral Triangle, it's a tiny part of the world's oceans. It's 1.6% of the global oceans, but it has 35% of the total global coral reef area. It's home to 76% of all coral species. It's home to 37% of all reef fish species. This is a biologically important area because Alfred Russel Wallace spent 8 years of his life there. And it was during his time in Indonesia that he developed the theory of evolution through natural selection. This has happened at the same time as Darwin. He doesn't get nearly as much credit, but this came out of the Coral Triangle. Another way to look at biological importance is that there are 363 million people that live inside the Coral Triangle. And more than 120 million of them depend on fish from the ocean for their primary protein source on a daily basis. This diversity supports the livelihoods of more than 120 million people in this region. If we look at the Philippines and Indonesia in terms of global fisheries production, combined, they are providing 7% of all of the fish that is consumed around the globe on an annual basis. The marine resources in terms of fisheries, in terms of tourism, this is supporting 8.4% of the gross domestic product in the Philippines and 4.8% of the gross domestic product in Indonesia. These are valuable economic resources for those 6 countries that comprise the Coral Triangle. And yet, the Coral Triangle is one of the most critically endangered marine ecosystems on the planet. So this graphic here, the red dots indicate coral reefs on the verge of collapse. The yellow dots indicate reefs that are not doing so well, and the blue dots indicate reefs that are doing okay. And what you can see here, very neatly, is the outline of the Coral Triangle. So the world's most biologically diverse marine ecosystem is one of the world's most threatened marine ecosystems as well. And despite the economic importance of these ecosystems, if you look in the Philippines and Indonesia, over 80% of these reefs are considered threatened. Despite the importance for food, for jobs, these are threatened. They're threatened because of coastal development. So people are living in high densities very close to the margins of the sea. Because of that, there's a lot of coastal pollution. So this is a beach in Indonesia with lots of plastics and trash. A relic of the US campaign in the Pacific in World War II is a practice called bomb fishing, where fishermen make bombs out of things that you can buy in the hardware store or old World War II munitions. And the shock wave from that explosion stuns the fish. They float up to the surface or they sink down to the bottom. And it's an indiscriminate form of fishing that basically kills everything within a certain radius. It also destroys the very foundation of the ecosystem that supports these fish in the first place. A growing threat is climate change. As the world gets warmer, so does the oceans. And these vibrant colors on the coral reef come from an algae that lives inside the coral tissues. And they're very temperature sensitive. So when the temperature gets hot, the corals get stressed and expel these algae, even though the algae provide 90% of their nutrition. And in many cases, the coral reefs cannot recover, and they get coated with algae, and they are lost basically forever. So, what can we do about this? What have we been doing about this? Well, before we get there, I'm going to start at the beginning, and not the very beginning, but at least the beginning for me. And we're going to start with the story. So this is a good way to start a story, from what I understand. And my story starts out in a place called Tucson, Arizona, a little town here in the desert. And I was a kid that loved animals, and I was surrounded by animals. And I've got lots of pictures like this. This is just the cutest one. And people always ask, so you grew up in a desert. How does someone that grows up in a desert end up being a marine scientist? And my standard answer to that is that when you live in a desert, you spend a lot of your time preoccupied with water. And so you're always looking for ways to get into the water. And one of the ways that I really started getting interested in marine science is through a television show that used to be on. See a lot of people of the right age group nodding their heads. The age group that knows a time when there were 3 television stations. And on one of those, on Saturday, I believe it was at 6 o'clock on Saturdays, they had the undersea world of Jacques Cousteau. And he and his crew went all around the world in their research vessel, the Calypso. Sometimes they would be looking at sharks, sometimes they would be looking at squid and octopus, or they'd be diving for sunken treasure, looking at the tragedy of the red salmon. But some of those actually went to the Coral Sea. The Coral Sea is in Papua New Guinea. It's part of the Coral Triangle. And it was through watching these episodes of Jacques Cousteau and having him bring this world into my living room that I got inspired to learn how to dive. And that set me on a path where I fell in love with the ocean, and is one of the reasons why I do what I do today. And one of the reasons why I spend so much time working with students trying to share my love and appreciation for this environment. The other show that I would also always watch was Mutual of Omaha's Wild Kingdom. I don't remember the phone number anymore. I have fortunately forgotten that. But it was hosted by Marlon Perkins with his trusty sidekick Jim Fowler from the San Diego Zoo. And it seemed like every other episode they were in some exotic place in Africa. And there was always a gratuitous shot of Jim hanging out the window. They called it a whirlybird. They never referred to it as a helicopter. And what he's doing here is he's about to use a tranquilizer gun to tranquilize a Cape buffalo. And I thought that this was so cool. You're like, tranquilizing animals, like hanging out of a helicopter. How cool is that? And it was because of being brought into the world of this show that I got interested in Africa. And as a graduate student, I spent a year in Kenya living in a tent along the side of a river, studying spotted hyenas. I did not have a whirlybird. That was cost prohibitive. I did have an old beat-up Land Cruiser. But I did have a dart gun, and I did get to tranquilize hyenas and put radio collars on them. And It was a really amazing experience. It was a project that didn't work out. If there's young people in the audience, either really young or undergraduate students, life takes a lot of different turns. Just take the opportunities that are given and things will work out. This was an opportunity that I took that did not work out. So I focused my PhD research instead on these little guys here. They're called canyon tree frogs. And canyon tree frogs, they're these cryptic amphibians that live on granite rocks on the margins of these small little isolated basins of water that occur in a sea of desert. And these little isolated pockets of habitat surrounded by a sea of desert are part of sky islands, which themselves are these isolated pockets of habitat where you've got this big, tall mountain range that rises up from a very flat, hot, inhospitable desert floor. And you can see where these sky islands are by looking at a satellite image, because all of these areas where there's this dark forest green, those are sky islands. And the interesting thing when you start looking at a map like this is that you can see it's like, all right, well, there's a sky island here and here and here, and the distance between these can be 30 miles, 40 miles, 50 miles, 80 miles, and we're talking about a small little frog that lives along the margins of these isolated basins of water surrounded by a sea of desert. And my thesis was trying to figure out, how did this happen? This is like really weird. These frogs are not going to hop across 50 miles of desert to get to a new habitat. And so I spent a lot of time driving around the desert with my trusty dog Shuka, hiking up into these places and catching frogs to my heart content. And then I would bring the samples back and I would look at the genetics of these. And what I found was that there were certain populations in sky islands that genetically were all very, very similar. And yet those were very different from other sky island populations that themselves were very, very similar. And the reason that you get this genetic similarity is because there's a lot of exchange of individuals among these populations. But it can't occur today, but when it did occur was back during the ice ages when conditions were wetter and cooler, and the habitats were much more continuous, allowing for dispersal corridors. Now, you guys should all be thinking one thing right now. What does this have to do with the fact that there are 80% of the reefs in Indonesia and Philippines that are threatened? And it's a reasonable question. So when we have a really special natural environment, what do we do? What we typically do is we create some sort of reserve or protected area. So here is a sign from a reserve. It's a game reserve in South Africa. You don't feed the monkeys, that's a good idea, and you stay off of motorbikes, and you don't get out of your car because of the large, dangerous animals. And people set up reserves in marine environments as well. So there's marine reserves or marine protected areas, also known as MPAs, and this is a sign from an MPA, also in South Africa, where you can swim and surf and fish. But you're not allowed to collect the oysters or the lobsters or bring shells home with you and things like that. Now, these reserves are actually quite effective in increasing the density, size, and diversity of the organisms that are inside. So inside of the reserve, you have many more different species. They are larger, and there's a greater density than outside. Another very interesting part about the impacts of marine protected areas is that there's a spillover effect. And if you have a marine protected area where you've got your increased diversity and density and biomass, and you go just outside of the marine protected area, what you see is that there's elevated densities and diversity there as well in comparison to populations that are even further away. And this is really important because when you create a marine protected area, there are certain groups of people, like fishermen, that get a little upset that they no longer have access to this resource. And the fact that you get this spillover effect and you get better fishing near the NPA is a really beneficial outcome. Now, despite their effectiveness, there's some limitations to using this model of terrestrial reserves in a marine environment. And to illustrate this, we're going to go back to Africa and go back to these hyenas. And this cute little hyena cub, when he is about 3 years old, and much less cute, that hyena cub will leave the clan of hyenas into which it was born and will disperse into a clan in a neighboring area. And when that happens, that 3-year-old hyena may travel 10 kilometers, 20 kilometers, 30 kilometers, but the amount of distance that it travels is much smaller than the scale of the reserve. So these reserves can be, hundreds of thousands of square kilometers. And so dispersing 20 or 30 kilometers in the Serengeti is no big deal because the Serengeti is really, really big. Now, how do things disperse in marine environments? Well, Pixar and Disney have done a great disservice in terms of our understanding of, or at least the general public's understanding of dispersal in marine environments. In case anyone, I hope I'm not bursting any bubbles here, but adult clownfish do not hop on the backs of sea turtles and get on the East Australian current to go from the Great Barrier Reef to Sydney. It doesn't work that way. So sorry to burst your bubble. In fact, most organisms in the marine environment as adults never move. They never move because, like these clams here, they're attached to the bottom. Or even mobile animals like this clownfish or this manna shrimp, they would never leave an area the size of this stage. And even some of the more mobile marine organisms would never leave an area the size of this room. And so how do you end up with reefs spread all across the 600 or the 6 million square kilometers of the coral triangle with the same species, even though those reefs are all isolated on little islands spread throughout the coral triangle? Well, the way that this happens is that it's not the adults that are moving, but a larval stage. So here are some lobster eggs, and the lobster eggs hatch, and the female lobster is fanning her pleopods here to get the larvae up into the water column. And these larvae are a few millimeters in size. And they can swim, but they're not great swimmers. And so what happens is that they disperse on ocean currents. So the currents make them drift away from where they were born. And this drifting on ocean currents can take them a very, very long way from where they originated. So what we're looking at here are some snails that live in coastal marine environments of West Africa. What these black dots represent are places in the Atlantic Ocean where Rudy Scheltema from Woods Hole Oceanographic Institute found the larvae of these coastal marine snails. So these, even though they originated in West Africa, these larvae can make it all the way across the Atlantic Ocean, thousands and thousands of kilometers. And in general, what happens if we look at how long a marine, the duration of the marine larval stage, so it can be from hours to days to weeks to months, but the longer the duration, the further the distance traveled. Now I'm going to repeat that because in Los Angeles I know that this is a confusing concept. That the longer you travel, the further you go, right? So the bottom line is that these larvae can travel for hundreds or even thousands of kilometers. And what that means is that if you're setting up a marine reserve, the larvae are not going to respect the borders. You can put a fence around it, following the terrestrial model, but what's going to happen is all the larvae that are produced on this reef here are going to go up into the water column, and they're going to disperse to some reef downstream. And the adults here came as larvae to this reef from some other distant reef. And so if you have an area that you think is really special and you want to protect these two areas here as coral reefs because they're beautiful dive sites or very productive fishing grounds, you have to know where the larval sources are. And those larval sources are going to be somewhere upstream. And if you do not protect the sources of those larvae, the larvae will never make it here. And you can protect that reef as much as you want, but if the larvae don't come in to replenish the mortality of adults, that reef ecosystem will disappear despite your best efforts to protect it. And this exchange of larvae among ecosystems is called connectivity. And this is one of the most critical gaps in our scientific knowledge, preventing us from doing better and more effective marine conservation planning. Now, how can we follow marine larvae through the environment? There are very few organisms that are conducive to radio tracking and radio telemetry. So big things like sea turtles or sharks, you can put on these sophisticated satellite tags that are constantly communicating their position. But most of these larvae in the ocean are the size of a grain of rice or smaller. And only one of maybe 100,000 survive. So even if you could follow one, even if that was possible to follow them through the ocean for 6 weeks in an environment where we don't belong, the odds of us picking the right one to follow is basically zero. So how do we address this problem? So to illustrate this, I'm going to talk about my mother. My mother had 4 kids. So that's Andy, Michelle, my younger sister, Lisa. We, in turn, have had kids of our own. And within our family, there's a genealogy. Within all of our families, there's a genealogy. And we think about genealogies enough that there are big websites like genealogy.com that allow us to track these things and learn more about where our ancestors came from. How many of you guys have ever thought about the genealogy of a clownfish? Anyone? All right, we have a couple people. All right, that's good. This clownfish, has offspring, and those offspring have offspring, and those offspring have offspring, and so on and so forth. And, there is a genealogy of clownfish. And at some points along that genealogy, when DNA is replicating in an individual, a mistake can be made. And that genetic error, that mutation, can be passed down to all of its descendants. So now we have 3 different branches of this genealogy here that come from these mutations. And we can see these mutations by doing DNA sequencing. We can see these genetic changes by doing DNA sequencing. And we can see it here where there used to be a C at this particular position, and now that's changed to a T. There used to be an A at this particular position, and now that's changed to a G. And so we can use DNA sequencing to look at this genetic variation within populations. And to make it simple, right now I'm going to walk you through an example where we're just going to look at two different types. And these genetic types we refer to as genotypes. And we have a blue one and a red one, type 1 and type 2. And so we can go out into a population and we can look at these clownfish, not necessarily as clownfish, but as genotypes. And genotype 1 and genotype 2 are an equal frequency in these two different populations. And we can summarize those equal frequencies by these pie diagrams here, where we have equal frequency of the blue and the red. And the only way that this happens in the natural environment is if larvae are moving back and forth between these environments, because the larvae move their genes, and it's the exchange of genes back and forth that causes these populations to become homogenized. Now, contrast that with this example here, where type 1 is only in population 1, type 2 is only in population 2. And the only way that this happens is if the larvae that are dispersing away from these coral reef environments cannot make it past some sort of barrier to dispersal. Now, what we've been doing for the last 10 or 15 years is using this genetic approach to understand how larvae are moving through the environments of the coral triangle. And we've done this through an educational program called the Diversity Project. And the goals of the Diversity Project is to understand the processes generating diversity within the marine environments of the coral triangle so that that information can be used to protect that diversity, while at the same time trying to increase diversity within marine sciences. And so we take students to places like Bali, where they learn to scuba dive and then get trained as scientific divers. And once they are scientific divers, then they can go out with us into the field and work with us doing the field collections, and then take those samples back into the lab where they learn modern molecular genetic techniques to reconstruct the dispersal history of these larvae through the coral triangle. And this has been a very successful project. We've had 42 alumni to date. Almost all of them are still in science. 70% of them go on to graduate school. Here are some of our recent success stories. Martha just graduated from Harvard a couple years ago. Alexis graduated last year from Santa Cruz. And next month, Melissa Kemp will graduate with her PhD from Stanford. So we are changing diversity in marine sciences while we are understanding diversity in the coral triangle. Now, what has the data shown us? Well, what we see is that there are similar genetic patterns across multiple species in the coral triangle here. So I'm going to walk you through 3 of the basic patterns that we see, showing you one example of many that we have of that pattern. And the first pattern that we see is divergence among populations in the Indian and Pacific Ocean. And we see this in small reef fishes and big pelagic reef fishes and snails, in clams and sea stars. And what this looks like, if we look at the data from the checkered board wrasse here, Indian Ocean populations look very, very different from Pacific Ocean populations. So on either side of the coral triangle, there is some kind of barrier to dispersal. Well, what could that dispersal barrier possibly be, given that most of the coral triangle is actually water? Well, this brings us back to the ice ages, right? Now, the ice ages were characterized by these big glaciers building up on land, right? So when those glaciers are building up on land, where is that water coming from? It's coming from the ocean. And so as glaciers build up on land, sea levels begin to drop. And the coral triangle begins to look like a very different place, where The Sunda Shelf here is completely exposed and is now a terrestrial tropical rainforest. The Sahul Shelf as well is also exposed and a tropical rainforest. And now we have a barrier between the Pacific and the Indian Ocean. So this is all due to history, much in the same way that those ice ages impacted the dispersal history of the frogs that I studied. But in the case of the frogs, it provided a dispersal corridor. In the case of the coral reefs of the Coral Triangle, it provides a barrier. The other pattern that we see a lot in mantis shrimp and clownfish and giant clams is divergence across a region called the Maluku Sea. This is the Maluku Sea right here. And what we see is that populations on the eastern side look very different than populations on the western side from a genetic perspective. And the reason that this is occurring is because of ocean currents. Now, what I want you to do is focus your attention here, and you can see this water spinning in this eddy. That eddy is called the Halmahera eddy. And what happens is that water from the northern coast of the island of Guinea travels north and to the west until it hits the island of Halmahera. And then it does a U-turn and goes back to the east on the Pacific equatorial countercurrent. And because the water from this region here isn't making it across the Maluku Sea, well, what's in the water? Larvae. And the larvae have their genes. So if the larvae don't move because the water don't move, the genes don't move, and you get divergence on either side of this physical barrier. Now, the third pattern we see is really easy because it's just a combination of the first two patterns. And that is that you have divergence across between the Indian and Pacific Ocean as well as the Maluku Sea. And we see this in, again, clownfish and stomatopods and clams. I'll illustrate this with an example from the boring giant clam. It's a boring giant clam not because it's unexciting, but because it actually physically burrows and cements itself into the reef. So here the populations in the Indian Ocean look very different than populations from the central part of Indonesia and the Philippines and populations on the other side of the Maluku Sea barrier. So if we integrate the patterns that we see across the 40, 50 different species that we've looked at to date, what it looks like is something like this, where we've got an Indian Ocean region, a central Indonesian and Philippine region, this region here of eastern Indonesia, and one of the areas I didn't talk about is Teluk Cendrawasih. It's also one of these areas that's genetically very different. And this is very valuable information to conservation planners because when you're trying to do conservation on the scale of 2/3 of the United States, you have to break it up into manageable chunks. And so that's what they do. They create a map of these marine management areas. And some of these barriers that we see here, some of these borders that we see of these marine management areas are actually coming from our genetic data. Now, one of the other ways that our data had a really big impact is that when they started this prioritization exercise, they were only focused on the Coral Triangle here. It never occurred to them to think about Western Indonesia. But when we showed them our data and showed how special these populations are from a genetic perspective, they've actually added in this region of Western Indonesia for prioritization for conservation efforts. So that's been a good development. Now, we've been extending the genetic work through collaborations with some very talented computer modelers, namely Jonathan Kuhl and Eric Tremmel. And modelers can do things that we can't, which is they can look at the distribution of all the coral reefs throughout the Coral Triangle region. And then with data from NASA and JPL, they can create a virtual ocean that they release virtual larvae into. And it looks something like this. And the model can have lots of different parameters, including the reef location and area, the ocean currents, how frequently they spawn, how long the larvae are in the water column, behaviors of the larvae, mortality. And what we're seeing here is larvae that are being released and dispersing, and we get to see where they end up. And you can integrate this over hundreds of thousands of generations and multiple species, and you end up with a connectivity map like this, where red indicates high connectivity, yellow is sort of moderate, and anything below that, it's essentially isolated. And what you see is exactly what we've seen in the genetics, which is the Philippines and the central Indonesia are one group. Eastern Indonesia is another group with this special region in Teluk Cendrawasih, the Bay of Paradise Bay. And then Western Indonesia, which got cut off here, is its separate group. So this is all very good. It's helping conservation planning in this region. But when I first started on this research project in the late 90s, early 2000s, I ran across a friend of ours who he was leading the formation of the Komodo National Park, including all of the marine environments as well. And using some very colorful language that's not appropriate for some of the audience here, he basically told me that I had no idea what I was talking about and that what I was doing was meaningless to conservation, because even if I could tell him where every larvae originated and ended up across the entire coral triangle, how is that information going to help him stop people from throwing bombs in the water? And he had a point. And I've thought about that a lot over the years, and realize more and more as I work there that our data is important, but it may be not the most important thing. And what I've come to realize is the lack of data isn't necessarily the most pressing problem. And how we can understand what the real problem is can be illustrated with these slides here. You can go online and you can buy shirts, coffee mugs, bumper stickers for saving the whales. You can buy books about saving the whales. You can watch really good shows about saving the whales. You can buy bumper stickers about saving the rainforest. You can go to protests about saving the Amazon. You can buy books, 50 Simple Ways to Save Rainforests. You can combine your love of rainforests with your love of cetaceans and buy bumper stickers of saving the Amazonian river dolphins. But if you do a Google search of Save the Coral Triangle, you come up with nothing. There are no bumper stickers. There are no mugs. There are no T-shirts. There are no rallies. And it's because of this. You protect what you love what you know what you're taught. And I got involved in this because I grew up watching shows like Jacques Cousteau and Wild Kingdom. And through these programs, they brought me into a world that a kid from the desert in Tucson, Arizona would never have experienced otherwise. And it made me want to go to these places and learn about these places. And that's why I do what I do now. And that's why I'm trying to pass on this knowledge to the next generation, including my own kids here, exposing them to these environments so that they can see how amazing they are, in hopes that the next generation will view these environments differently than many of the current generation. Now, Indonesians are closely tied to the sea. They pray at the sea, they fish in the sea. Sea is an important part of their recreation. But Indonesia is the world's 4th largest country, 240 million people. And what you see here is an extraordinarily rare sight. It is an Indonesian scientist who is a scuba diver. There aren't very many of them. In fact, there'd be a lot more places for the people in the back to sit if the only people in these seats were Indonesian scientists who were scuba divers. And because there aren't very many Indonesian scientists that can scuba dive, in fact, most Indonesians, it's a country of 17,000 islands, most Indonesians don't swim. And because of this, they've never seen what's beneath the surface of the water. They've never seen these amazing habitats. They've never seen the diversity that's there or the impacts that the populations in the Coral Triangle are having on these environments. And going back to this African proverb, you protect what you love what you know what you're taught. If they are not learning about these environments, if there is no one to go out there and study these environments and teach about the importance of these environments to other Indonesians or Filipinos or people from Papua New Guinea, you're not going to have the necessary inertia within the society to make a change. And another big problem, so you can really see the impacts of this problem with this graph here, and I promise this is the one and only graph that you'll see in my talk. And what I did is I just did a literature search from a couple of journals that people would commonly publish marine literature in. And I searched for publications on the Great Barrier Reef going back to 1989. Here we've got numbers of publications per year. And this is what it looks like for the Great Barrier Reef. People have heard of the Great Barrier Reef? Yeah, exactly. Nice place. This is what it looks like when you do the same thing for the Coral Triangle. Now, don't get me wrong, Great Barrier Reef is a nice place. But the Coral Triangle is the global epicenter of marine biodiversity. It's economically incredibly important. And it's one of the most threatened marine environments on the planet, and it's also one of the least studied marine environments on the planet. Now, what's the impact of this? It's actually not what you think. It's not the data. And this is what I learned from my friend. It's not about the data. What it's about is this. Every time you pick up Discover magazine or National Geographic and you read an article, every time you turn on the Discovery Channel, Animal Planet, you look at a blue chip nature documentary on PBS, those reporters, those journalists, those executive producers that are making these media products, they're not coming up with these ideas on their own. What they do is they read the literature. And I know this because my wife, Jeannie, in the front seat here, worked for a scientific documentary production company, and she read the literature probably as much, if not more than I did, looking for interesting stories. And so that's why we thought, all right, if we're going to really do something about conservation in this part of the world, we need to take a different tack. And so with the help of some very dedicated collaborators, both in the US and in Indonesia, 4 years ago, we founded the Indonesian Biodiversity Research Center. And it's a collaboration between Udayana University, Diponegoro University, the State University of Papua, UCLA, the Smithsonian Institution, Conservation International. It's funded by the US Agency for International Development. And the goal of this center is to develop the Coral Triangle into an international hub for biodiversity research. So we have leading scientists from around the world come out there and teach short courses to promote biodiversity research. Building on the model of the Diversity Project, we start by teaching them scuba. In some cases with Indonesians, we start with swimming. They have to do that on their own before we show up. They got to do a little homework. But then we bring out some just world-renowned scientists. So this is Nancy Knowlton, who's the Sant Chair for Marine Sciences at the Smithsonian Institution. She's a member of the National Academy. She's the head, or was the head of the Census of Marine Life. And we get these amazing scientists to come out here and teach at the center. And we teach the students how to study biodiversity. So Nada in here is taking all of the animals off of a dead coral head, and they put these into individual cups. And then through traditional taxonomic techniques, they try to identify what's there. And we build on this by not just looking at traditional taxonomy, but we teach them modern molecular genetic techniques so that they can take a genetic approach to documenting and understanding biodiversity. And this center is promoting the success of students here at Indonesia, or at UCLA. So here's in the front row here, a bunch of students from my lab and students from the Diversity Project. That's Ketut, the driver. I think he took them to their village. But it's not just about the US students and the UCLA students. It's about the Indonesian scientists that we are developing. So I'll just give you an example of a few here. So this is Aji Anggoro. He just received a fellowship from the Indonesian government to do his PhD at UCLA. And we'll be using these devices called autonomous reef monitoring structures that you put out onto the coral reef. And they get encrusted, all those larvae, they land on it, and they make it their home. And you get these, these plates encrusted with different organisms, and you can identify them. And we're doing this with molecular techniques, so we can look at diversity from viruses to microbes to these animals here. And these structures can be used to monitor the impacts of climate change or the effects of pollution and see how diversity is impacted by these human activities. Rita Rakhmawati, who is also in the audience here, she's a PhD student with me right now looking at regional variation in coral bleaching across Indonesia, because there's some places that seem to be immune or relatively immune to coral bleaching, and she wants to figure out if there's something we can learn about those environments to make the areas that are very sensitive more resistant to coral bleaching. Dion Pertiwi is in the audience tonight. She's visiting our lab from the IBRC. And among many of the projects that she's doing, she's using next generation sequencing technologies to try to identify management areas in commercially important tuna. And I'll finish with a important success story highlighted by Andre Sembering, who was a student at the IBRC. And there's a big trade in shark fins, a lot of which are illegal. But when they're in a big bag like this and all dried and they're disassociated from the animal that it came from, they're really hard to identify. And so he and others at the IBRC wrote a proposal, got it funded, to use genetics to identify these shark fins. And they found two very disturbing results. The first is that there are almost no reef sharks represented here, and that's because they've been almost completely fished out. And instead, the fishermen now are targeting things like scalloped hammerheads, which are CITES Appendix 1, but the Indonesian government didn't even acknowledge that they existed in Indonesian waters. And so there's actually new legislation being considered in Indonesia to protect sharks, certain species of sharks, based on the data that's being generated at this institution by people who didn't have these skills 4 years ago. And so our goal with this center and our educational activities in taking this different approach to doing research where, we need the data, the data is important, but what's more important is how we get the data, because we can get that data in a way where we can train and inspire a new generation of marine scientists. And if we do this effectively in the Coral Triangle, what will happen is that rather than seeing this flat line here, we will see a lot of growth, and that will bring in the reporters and the film crews, and that's going to grow the public consciousness so that one day, hopefully soon, on the streets of LA, I might see something like this, Save the Coral Triangle. And it's once that we grow that public consciousness, once I can go into a room like this and at the beginning of the talk, everyone can raise their hand and say, I know what the Coral Triangle is and know why it's important. That's when we're going to be able to change the fate of the Coral Triangle and have reefs that don't look like this, but look like this, and are there both as sites of natural splendid beauty, but allowing that diversity to persist so that it can provide the food, the jobs, and the economic benefit on which hundreds of millions of people depend. Thank you.